You know, in the world of IVF, there are some rules that have always felt absolute. Certain embryos are just deemed abnormal, non-viable, with zero chance of becoming a healthy baby. But what if one of those rules was just completely shattered? We're about to dive into a groundbreaking case that really challenges everything we thought we knew about the limits of life. So that's the big question here, right? Can an embryo that everyone considered a lost cause actually be saved? It's a story that starts with what's normally a devastating diagnosis, a fatal error in fertilization. But believe me, it ends with an outcome that could change everything for some families. Okay, so to really get why this case is such a huge deal, we first need to understand a common and honestly often heartbreaking part of the IVF process. What happens to these so-called abnormal embryos?
Think of it like this. When the genetic material from the egg and sperm combine, it's this incredibly complex dance. And sometimes, errors just happen. An embryo might end up with the wrong number of chromosomes, or like in the case we're looking at, an error happens at the very, very first step.
And for these embryos, the protocol has been brutally clear for decades. They're considered non-viable, they're linked to failed implementations, miscarriages, or serious genetic conditions. The standard procedure is to discard them. End of story. And that's been the accepted medical reality until now. So what was the specific error in this case? Well, it involves something called a 3PN embryo. Let's break down what that actually means, because it is right at the heart of this entire story. All right, imagine the genetic material from each parent is like a blueprint, an instruction manual. After fertilization, you can actually see these two little packets of instructions. They're called pronuclei inside the single-cell embryo. A healthy, normal embryo is supposed to have exactly two of them, one from the egg, one from the sperm. Simple enough, right? A 3PN embryo, then, is one that shows up with 3 pronuclei. It's got an extra blueprint. This has always been a massive red flag, a clear sign that something went seriously wrong during fertilization, and it's always been considered a fatal error. But what if? What if you could just remove the extra copy? And this idea is just so brilliantly simple. The researchers who pioneered this work described the concept this way, and I absolutely love this analogy. If you have a document with a duplicated sentence that's making it unreadable, you don't throw out the whole document. No, you just delete the extra sentence. Okay, but how on earth do you perform surgery on a single cell? I mean, this sounds like something out of a sci-fi movie. This is where incredible skill and microscopic precision come into play with a procedure called pronuclear enucleation. Now check out how methodical this process is. First, the embryologists have to spot that 3PN embryo. Then, and this is the wild part, they use these incredibly fine instruments to literally perform surgery and remove that extra pronucleus. After that, it's not over. They have to culture the embryo for several days just to see if it develops into a blastocyst. And crucially, they do genetic screening to make absolutely sure it's chromosomally normal before they'd even think about a transfer.
And you gotta understand, this wasn't just some random experiment. This was a last-chance effort for a patient who had very few options. Out of 7 of these 3PN zygotes, embryos that would have otherwise just been discarded, the team attempted this rescue procedure on 6 of them. This was truly uncharted territory.
So, we have the theory, we have the procedure, and we have the patient. Did this audacious microscopic rescue actually work? Let's look at the results. So, from the 6 embryos that they operated on, 4 didn't develop correctly, but 2 of them developed into high-quality blastocysts. I mean, that alone is a remarkable result, turning 2 supposedly non-viable embryos into potentially viable ones. After the genetic screening, one of those blastocysts was confirmed to be chromosomally normal, and it was transferred. The result of that transfer? Well, it can be summed up with this one powerful number. 1. One healthy, full-term baby boy, a live birth from an embryo that, by all existing standards, should have been thrown away. So the crucial point here is that this single success story suggests our entire definition of non-viable might need a major update. Now, as exciting and mind-blowing as this is, the researchers themselves are urging a ton of caution. And they're right. A single success story is just not the same as a proven medical treatment. It's absolutely vital to balance the incredible hope this offers with the scientific reality.
And this slide lays it out perfectly. On one side, the promise is immense, a potential rescue technique for patients with no other options, a discovery that could one day change IVF protocols for everyone. But the reality is that this is highly experimental. We have no idea about the long-term safety, and the procedure can't even guarantee a fix. This is not and should not be considered routine practice yet.
This one birth has basically kicked open the door to a whole new frontier in fertility science. But with that comes a wave of new and really critical questions that we have to answer before moving forward.
The path forward is clear, but it has to be careful. Right now, the immediate next steps are to refine these surgical techniques, and most importantly, to conduct much larger studies to assess safety and see if this really works consistently. In the near future, scientists are hoping to figure out if some 3PM embryos are better candidates for rescue than others. And long-term, the ultimate goal is to understand what causes this error in the first place, so maybe we can one day prevent it altogether.
So where does all this leave us? Well, we've seen that the seemingly impossible might just be possible, that a 3PN embryo can, in very specific circumstances, be rescued. But it's so important to remember what we're looking at here. This is an experimental, last-chance technique. Its success hinges on immense expertise in screening. It is a powerful glimmer of hope, for sure, but it is not a cure. And those big questions about long-term safety and whether this can be done again, those are still very much unanswered.
This single case forces us to reconsider a long-held biological absolute. It's a powerful reminder that science is constantly evolving, and it leaves us with a final, fascinating question to think about. What other fundamental rules that we all take for granted are just waiting for the right discovery to come along and be completely rewritten?